So, tonight our first speaker is Michael Purugganan, and he's inextricably involved in this GMO controversy, as you'll hear. I think you have an exciting night this evening, in part because Michael is a native of the Philippines, and he has a passion for both journalism and science. Now, during his undergraduate days at the University of the Philippines, he reported for his college newspaper during the events that led to the downfall of Ferdinand Marcos. He later worked as a news stringer for Time, Newsweek, and Associated Press. All of this while he was still an undergraduate. Now he got a master's degree at Columbia, as I recollect, and he earned his PhD in the U.S. at the University of Georgia, working on transposons with Susan Wessler, one of their very first postdocs. And he made it clear in no uncertain terms that I had to acknowledge him as another of my grandchildren, albeit intellectual. He is currently the Dorothy Schiff Professor of Genomics and Professor of Biology at New York University, and he has served as Dean for Science since 2012. He spends time in our part of the world as well. He's on the faculty at NYU Abu Dhabi, and is the co-director of the Center for Genomics and Systems Biology at that NYU Abu Dhabi Center. His research interests are in plant molecular evolution, including the role of plant transposable elements, the diversification of regulatory gene families, evolution of development, molecular population genetics, domestication of crop species, and microbial social evolution. Michael has already accumulated a bunch of awards and honors, including the Alfred Sloan Young Investigator Award back in 1997, Guggenheim Fellowship in 2006, Sigma Psi Prize, and he's also, if I recollect, a fellow of the AAAS. Michael.
So, first of all, thank you very much, Nina, for that kind introduction. And I really, Dr. Fedorov is my intellectual grandmother. And so it was really a pleasure to accept the invitation to come here. And also I'd like to acknowledge the organizers and also Mark Tester and Sonia Negrell for inviting me to give a talk in this really very interesting and exciting event. And it's really, it took me like 3 seconds to decide to come here just because, for two reasons. One was that I thought, wow, what an exciting event to come to. And also I had all of these connections here at KAUST. And also I was intensely curious about KAUST because, of course, I'd heard about it all this time. And it's wonderful to be able to see the campus, to see all the energy, and to see all the research that's being done. I'm really quite overwhelmed by what I'm seeing. As Nina said, I work on a large number of areas of research in my career, from plant transposable elements where I began, and that's why I'm also Dr. Fedorov's intellectual grandchild, all the way to a lot of my interests now are the evolution of domestication. And today in this sort of series of keynote lectures, we're going to talk about the past, the present, and the future. And Dr. Ingo Petrikas is going to be talking about, after me, he's going to be talking about the present and the future. Well, I'm going to be talking about the past. And the reason for that is, I think by looking at the past, there are some lessons we might be able to learn about how to feed the world. Now, we know that plant genomics has been an important tool in the last few years in the study of plant biology and the study of all of biology. And for us in this series of events here at the Winter Enhancement Program, we're acutely aware about the need to feed a hungry world. Dr. Fedorov talked earlier this afternoon about the large population increase and the many challenges we face. And there are a lot of challenges we face in order to be able to feed a hungry world. We talked about increasing population, the land pressure that we observe, and of course climate change, that require in part creative technical solutions in order to solve some of these problems. And plant biology, plant genomics is one part of the possible solutions that we might be able to use to address some of these issues. One of the issues that, of course, is on our minds as we move further into the 21st century is the fact that the climate is changing. We are now seeing an increase in global temperatures. That has been documented. The projections of increase will continue. And this is causing changes in the climate both globally and regionally as well as more erratic climates that we're observing from season to season and from year to year. And this is a challenge that we're all together on this planet, whether we're here in Saudi Arabia or the Middle East or around the world, are going to be facing. This is the starting point of my talk, and although this is some of the problems we face, I'd like to note something interesting. The last time there was a global warming episode after the last glacial cycle was when man began the process of domesticating crops and animals. Okay? The domestication process by which man, or humans, I should say, took plants from the wild and started cultivating what is thought to be, although this has not really been proven, is thought to be partially in response to the warming of the globe about 10,000 years ago, which changed climates regionally and allowed for cultivation to occur in order for humans to get a more stable supply of food throughout the year. And so these Neolithic peoples, this point in time in our history, which has been called the Neolithic Revolution, was a time when humans took wild species, which previously they used to acquire food by hunting and gathering, and brought them into cultivation. And you can see here archaeological digs of some of those earliest Neolithic sites, whether it's in the Jordan Valley, of course, near here in the Arabian Peninsula, starting 9,000 years ago. This is one of the earliest Neolithic sites. It's known to contain signs of domesticated crops and fields, or this Neolithic settlement in China. But throughout the world, actually, in different parts of the world, we see the rise of agriculture and the domestication of crop species. And as I said, this enabled societies around the world to begin to feed greater and greater numbers of people. It did contribute to the rise in population pressure. It caused populations to expand because it allowed societies to feed people throughout the year in a more stable system. Why I'm interested in evolution, aside from in domestication, aside from what it might tell us about how to increase plant yield and to help in some of the challenges that face us, is I'm a molecular evolutionary biologist. I'm interested in the evolutionary process. And many of you might not know this if you've read The Origin of Species by Charles Darwin, which was published in 1859. And we just celebrated the 150th anniversary of the publication of this book a few years ago. The first chapter in Origin of Species is all about domestication, or domesticated species, because Charles Darwin, in formulating his theory, his discussion on evolution and the theory of natural selection, drew on the variation he observed among domesticated species as a way to tell people the importance of variation in the process of evolution, and also used the process of selection that humans applied to domesticated species as an analogy to natural selection, which he thought of as the major force that drove the evolution of species. And the first book he published after Origin of Species was the variation of animals and plants under domestication. He was really, truly fascinated by the domestication process. And so in my laboratory, we've been interested in domestication for what it can tell us about this process of evolution. So domestication is an interesting evolutionary process. It's an interesting evolutionary process for several reasons. One, it's a very recent evolutionary process. All domesticated animal and plant species, with a possible exception of the dog, was domesticated only in the last 12,000 years. Okay? So every species that we cultivate, every species that we herd, every species that we use as pets, only arose in the last 12,000 years. Because of this, as an evolutionary biologist, we think that we can still see the signature of this process of evolution in these species and in the genomes of the plants and animals that make up the domesticated species. So we think of them as a model for evolution. What are domesticated species? It's really, these are species, it's very hard to define a domesticated species, but probably one of the better definitions are, these are species whose reproduction is assured by humans because they meet human needs in a variety of different ways. So this range from crop and ornamental species to livestock and pet species. A very conservative estimate says there are about 250 plant species that have been domesticated and 44 land animal species. I've seen estimates that are one order of magnitude higher than this, but I think this is probably a good place to start. And the domestication process, at least in crop plants, remember, is a process that involves two players. It involves the plant, that's whose evolutionary trajectory we are changing because we're bringing it into cultivation. And it involves the human partner. Humans changed their behavioral ecology starting about 12,000 years ago in how we fed ourselves. So prior to 12,000 years ago, how we fed ourselves was largely by hunting and gathering. But about 12,000 years ago, human behavioral ecology changed. So we started to cultivate plant species. And that changed the environment of the plants, changed the selective pressure on these plants, and led to the development of new species that we now call domestic species. There are at least 24 regions around the world where domestication occurred. We know that they occurred in many of these places independently. One of the reasons we know that is that it occurred independently clearly in the Old World and the New World, which had no human contact prior, starting about 15 to 20,000 years ago. The earliest evidence for domestication and the rise of agriculture occurred, of course, very near here in the region called the Fertile Crescent, which spans everywhere from the Levant all the way to southern Turkey and into Iraq. This has the earliest traces of agriculture and domestication in the world. But other early examples of domestication are known. For example, in China, they probably domesticated plants independently. And also, as I said, in the Old World, especially in Mesoamerica and the Middle America, there's very early signs of domestication as well. So domestication was a global phenomenon that involved human societies initially that may not have been really in strong contact or not in any contact at all with each other. And yet, from this, we have a diverse array of cultivated plants and animals that now are dependent on humans for their reproduction, and we use for a variety of needs for food, for clothing, for shelter, and so on. The stages in domestication can be fairly complex. It starts really with the differentiation of the wild and cultivated species. As humans begin to look into wild populations, take a subset of those and bring them into cultivation in the environment. That is, we start to plant them, we start to harvest them. As we start to do this process, that is when domestication starts to kick in. You need cultivation in order for domestication to proceed. And then you have increase in frequency of alleles that are desirable as cultivated populations begin to get established. And then as cultivated populations spread throughout the world, you start to see evolution of different varieties that are adapted to different local environments or even local cultural preferences. And then we're now at the final stage of the domestication process, where humans are now either deliberately breeding domesticated species or using genetic modification technologies in order to change the trajectory of evolution of these different species. I'm not going to talk really about this part of the domestication process. I'm going to talk about these parts, the early parts of the process of domestication. And over my career, I've studied several different domesticated species with various, in various types. I've studied, cauliflower, I've studied melons, corn was when I did my PhD, rice, I've studied barley a little bit, and I'm going to tell you especially stories around rice. So one of the things that we're interested in is the nature of speciation. I'm going to tell you what I'm going to do in this talk. I'm going to tell you a few stories about how the study of how domestic species evolve and how we can learn about how these interesting group of species have evolved. The nature of speciation is one area of interest. We know that one of the consequences of domestication is that the selection in domesticated species have led to very extreme phenotypes. Whether it's the ear of corn, which is a very radical departure from the small rachis of hyacinthe, or if you look at dogs and bulldogs, they really have very different morphologies, in some cases from wolves, which are their ancestor. Because of the selection for extreme phenotypes, there has always been a question for how fast the process of evolution under domestication has occurred. Because these phenotypes are so extreme, there has been a number of models of the process of evolution in domestication that say they occur very quickly and very rapidly. These are just a compilation, an old compilation, but it still holds, of models of evolution of various types of species. And what you want to see here is this column here, which has the pace of domestication. Rapid, 7 years. Legzinski thought that domestication of legumes took only 7 years as a process, 130 years. Most of this, except for only 2 models, suggested that they were very rapid. The domestication process occurred very, very quickly. And this was something that one of the things I needed to do as an evolutionary biologist to really test whether this was true. And the reason I had to test whether this was true is that my colleagues in evolutionary biology have always said to me, although less so now, that we really can't learn much about evolution by studying domesticated species, because the rapid evolution of domesticated species is very different from the more gradual evolution that occurs under natural selection for a large number of wild species. And so several years ago, I thought, is this really true? Does domestication in domestic species actually occur very rapidly? And I was fortunate to get to know an archaeologist at the University of College in London, Dorian Fuller, who's probably one of the world's expert on the archaeology of domestic species. And he has a really encyclopedic knowledge of how domestic species have evolved throughout the world. And in conversations with him in London when I was visiting him, we realized that there is a fossil record for domestic species, that the archaeological record provides us with a fossil record that shows us how phenotypes have changed over time and to quantify the rate at which these species have evolved over time. So archaeology gives us a paleontological record of domestication. And so what we did a paper we published several years ago. We looked at the database that he had collected of records of different types of species throughout the world. We looked at the increase in the sizes of the seed and grain. We looked at the percentage of shattering, which is how the percentage at which the ability of plants, of non-shattering, the ability of plants not to drop their seed on harvesting has evolved. And one of the things, and this is a paper that's now we published a paper in Evolution a few years ago, but now we have another paper in press, is that the archaeological record is clear. It takes about 4,000 generations, or 4,000 years for domestic species to evolve. And 4,000 years, depending on how you look at it, can be rapid or slow. We think it's actually a normal pace for the evolution of most species, including wild species. If I compare the rates of evolution, and if you want to look at our paper in Evolution in 2010, where we compared the rates of evolution of domestic species with wild species, we find that the rates are actually fairly similar. So domestic species, the process of domestication, is not evolving, at least from the archaeological record, any faster than wild species are evolving in the wild. And so we think that this is, in fact, a very good model for the study of evolution. And again, I said we've updated this with data across multiple species looking at a variety of different traits. And most of the species, by the way, are cereal crop species that are found here in the Near East, in the Middle East. And if you look at the rates of evolution, if you look at the distribution, you find that much of the percentage of change per year based on the archaeological record for several different traits occur at the rate of 0.01 to 0.02, 0.01 to 0.025% per year, which is not a lot. But of course, over several thousand years can cause radical changes in many crop species, not all crop species. There are certain crop species that have clearly evolved very quickly, including the brassicaceae and maybe and corn as well. But for a large number of other species, it's evolving quite slowly. And we think it's because that much of the selective pressure that occurs in domestic species, we think of them as conscious selection or artificial selection, which can be very rapid and which causes the breeding of things like bulldogs, for example. Certain traits are clearly under conscious artificial selection. Fruit and seed color, fruit size, taste and smell are clearly things that are under artificial selection. But we think that a large number of traits, at least initially in domesticated species, are by unconscious selection that is really natural selection, whether it's the loss of seed dispersal, increase in seed size, height, and so forth. Early on in the process of domestication, these were really natural selection caused by humans putting these things within the context of an agricultural environment. So we think that human-associated domestication is really a natural evolutionary process that involves two partners. If you think of an evolutionary process that involves a plant and an animal, say a hummingbird and a plant that it pollinates, that's an example of a natural evolutionary process. We think humans planting plants is also a natural evolutionary process. It involves an animal-plant interaction. We're the animals, the plants are the crop plants that we are now planting. Okay? So what I'm going to talk about are, in looking at the evolution of these domestic species, there are several questions that also arise. For example, what are the genes that underlie these domestication events? How often have these species evolved? Have they evolved once or multiple times? And in the last few years, we've of course been helped, and this has been now known for decades, the advent of molecular technology has allowed us to sequence genes or genomic or small regions of the genome and get variation data that tells us how different strains or different cultivars or different individuals may differ from each other in terms of nucleotide diversity. Or, of course, with new technology, we can look at entire genomes and look at variation across entire genomes in different individuals or different species. And there's a large number of techniques that were developed the last few decades that allow you to take this data, molecular variation, and model the evolutionary process and infer what has occurred in the past in many of these species. And that's what I'm going to talk about today. So I'm going to talk about one story that we did several years ago. And that had to do with the origin of domesticated rice, which is a large part of what my lab does. We're interested in domesticated rice. It's a primary calorie source for a large portion of the world's population. It's inbreeding. It's an annual plant. And I'll talk to you about the fact that we think it has one domestication event. The wild species that gave rice, domestic rice, is Oryza rufipogon, which is found in southern Asia. It's an outcrossing or perennial and annual ecotypes of this species. And of course, Ingrid Patricus is going to talk about golden rice, which is probably the next stage in one of the next stages in the development of rice. What I'm going to talk about is what's been happening in the past. If you look at different rice varieties, and this is just something we did several years ago where we took data from 110 genes around the genome and used them to cluster them according to genetic similarity, you find that rice generally has two distinct genetic lineages. There's a lineage that includes a group of rice called the Japonica rices. These are the rices, for example, that you think of in sushi. And they are genetically related to each other. Aromatic rice falls into this category. And the Indica and Aus rice, Indica rice is the rice that you find, for example, in Saudi Arabia and that you use in Saudi cuisine. This rice looks like it's genetically distinct from the Japonica rices. And this has been known for some time. We didn't discover anything new when we looked at this. The fact that there were two distinct genetic lineages of rice had been known for some time from a variety of different sources. And because rice appeared to be composed of two genetically distinct populations, it had been thought that the way rice had evolved was the following. Wild rice, or Oryza rufipogon, was spread throughout South and Southeast Asia. And then about 9,000 years ago, Japonica rice evolved probably in southern China. And that at about the same time or soon after, Indica rice evolved in the Ganges Valley. This was a case in which people thought there were two origins of a domestic species. And so that had been what was the accepted paradigm for the evolution of rice. We had come, and this is something we accepted in the lab as well, in my lab. After that project that I just showed you, we then went on a project where instead of doing 110 genes, we sequenced a very large number of genes in 3 rice chromosomes, chromosome 8, 10, and 12, both for a large number of different rice varieties as well as wild rice. And this is what some of that data would look like. These are just the SNP patterns, or single nucleotide polymorphisms, across the 3 rice chromosomes, how they vary across these 3 rice chromosomes. And so we had a large amount of data from a large number of different rice successions for these 3 rice chromosomes. So this is where using that data and employing it in creative ways comes in. We were able to take that data and model the evolutionary history of rice in what's called a diffusion approximation that's based on the neutral theory. And the diffusion approximation is in this scary-looking formula down here that basically tells you the trajectory of SNP evolution or the evolution of these different mutations. And when we were developing this, basically you have to, in order to do this, you have to specify what is this history that you're looking at, the model of the history you're looking at. And what you do is you try to fit the data, which is the variation data in SNPs, into this model. And of course, what we were looking at was we thought rice evolved twice. So this is the model we were testing, and just so that you know what we're looking at, this just shows you the population of wild rice evolving over time. This goes from past to present. And then what this says is that sometime in the past, there was a population that evolved and that broke off, and this gave rise to Japonica rice. And then sometimes separately, another population broke off, and this gave rise to Indica rice. And then we're able to show, we're able to constrain this by how much time is evolved in these events, and also how much, not migration, but gene flow that is crossing between these different groups has occurred over time. So this is the model that's been accepted for rice evolution. What we just wanted to do was fit and find the numbers for all of these parameters in the model here. In order to test this, we decided we decided to do this just as a comparison control. We decided to come up with a model of one origin of rice. That is, it occurred once, and then an indica, or japonica, it really didn't matter, evolved from an already domesticated rice species. We didn't think this was right, okay? We thought this was right. This was just something we just threw in, a simpler model just to test this model. And so when we ran this analysis, and I'm not going to go through what we did, we took the frequency of SNPs in the genome. We ran what's called the site frequency spectrum. We have our observed. We look at our various models and what it gave us. And what you have to look here is this slide here, where it tells you the log likelihood. How likely is it that you get one model versus another model? And the more positive your likelihood, the more you accept the model. And if you look here, this is a single origin model. Okay, this is the double origin model. This is the model we thought was right. Look at its log likelihood, minus like 1088. Look at the log likelihood of the single origin model, 1038, 1054. And actually, that's significant. That difference is significant. It seemed like our model that said rice at one origin was outperforming our model which had rice at 2 origins. And the way we would do this was a collaboration between my group, a group at Stanford, Carlos Bustamante, Barbara Shaw at Washington University. We had a big lab meeting. Carlos was the one who actually presented this data, and my first reaction was, Carlos, you're wrong. Your model is incorrect. This can't be right. Okay, I don't believe this is correct. Go back, retune your model, put in selection, do something, but this can't be correct. It was fortunate that my postdoc at the time was leading my rice project, Jean Molina, came to me and was not listening to me. She came to me and said, Michael, why don't we go and look at the past data that said rice at 2 origins and reanalyze them? And I remember telling Jean, don't waste your time. Clearly, Carlos is a smart guy. He won the MacArthur Award. He's a certified genius. His model is wrong. This can't be right. But Jean persisted and didn't listen to his advisor, which was good. Because what she did was she went back to the literature and looked at different studies that looked at the phylogeny of rice and had come to the conclusion that rice had originated twice. And it turns out that all of these studies were wrong. And they were wrong because what they did was something that has just been shown to be wrong when you do these types of analysis. What all of these studies did, and what in fact we also did in an earlier study, was you take all of these genes and you treat them like they were one gene, and you analyze them together. And the reason you do that is that the information you have in each gene might be so small, because these are species that are evolving very recently, that you want to maximize the amount of information by putting all of the genes together and treating them like one. And it turns out that people had just done a simulation like 5 years ago and showed that if you do that, 75% of the time you're going to get a wrong answer. And indeed, every single one of these studies got the wrong answer, because we use a different method to analyze these previous data sets that had just come out. It's a Bayesian method to look at phylogenies in trees. This is a new method that takes all of the data for genes separately and integrates them later on. And in all of these methods, most of these methods, especially those that had a large number of genes that they were using, said that the single origin model was supported most of the time. Okay? So it seems that even those studies that had data that said rice had two origins were wrong, and that rice in fact had just a single origin. And in fact, that jives with the archaeological record, because if you look at the archaeological record, and what we thought here is that where rice really originated was China, because China has the oldest archaeological record for large-scale rice culture in the Yangtze Valley. If you look at where you can have clear evidence of cultivation, it started about 9,000 years ago in the Yangtze Valley. And that's where you have clear, these are archaeological sites throughout China where rice has been seen. But if you look at India, it's actually hazier, because the early evidence for the cultivation of rice in India was really only about 4,500 years ago. It was clear they were doing something, but it was not large scale. Rice just began to expand in India starting about 4,500 years ago. And it's associated in the archaeological record with the rise of pottery and implements that clearly had a Chinese influence. Those two things started to rise together. And so this gave us an idea for how rice had actually originated. Oryza rufipogon came, Japonica evolved first. In India, what they were doing was they were either harvesting wild rice or they were doing something, but it had not yet been domesticated. It was at a fairly low level. Japonica evolved and moved throughout East Asia and Southeast Asia, and then it went to India about 4,000 years ago, where it probably introgressed with either wild rice there or whatever they were doing with Proto-Indica, and that gave rise to the Indica rices that we now enjoy, for example, here in Saudi Arabia. And the archaeological record and the molecular record agreed with that, and the Beijing Genome Institute also published a paper 2 years ago in which they confirmed the results of our study by showing that domestication genes also support a single origin model for rice. Why this is important is because in order to do things like mapping genes that are important for yield or for looking at traits in rice, you have to also, in some of the newer ways of mapping genes, specify the model by which these populations are related to each other and their evolutionary history. What we're finding now is that, at least in rice, and now we suspect in other domestic species, that the evolutionary process is much more complicated than we thought. They may involve multiple domestication events, drastic changes in population sizes, and a lot of introgression and migration that goes on. And so this question of single or multiple origin of domestic species is something that crops up in the study of domestication over and over again, partly because when you look at domesticated species, some of them are so variable, you can't believe that they originated once. But it's clear that in some cases they originated once, such as dogs, but in some cases, such as the brassicas, they originated multiple times. So both things are occurring in different aspects of the evolution of these species. And by the way, one of these side notes that happens is that you get a lot of press and in very different ways in different countries that we've talked about. So this came out in our study. The BBC was very neutral about that. The South China Morning Post, which is in Hong Kong, was very nice. Your study backs China in Great Rice Debate. I love that. A row has flared after a study of the University. I love this one. Jakarta, which seems to be siding with India, was saying, U.S. and origin rice spikes Sino-India row. And Nina knows, she was the State Department, when you see the word Sino-India row in something, you get really nervous rapidly. But this was kind of fun. But as I said, what it's telling us too is that the models for evolution for these domesticated crop species are probably occurring in more complex ways than we thought. Okay, so that's just one aspect of how you use molecular data to look at the evolutionary history of these species. We now know that it's making us rethink for how these crop species evolve and that the process of domestication can have complex evolutionary histories that we need to revisit by careful modeling. Because there are really sophisticated methods that are now available to look at this data, especially from the human genetics community, and we're just applying them to looking at crop plants. So the other thing that we've always been interested in is the genetics of domestication. We know that all of these great traits have evolved during the domestication process or in the process in which plants are moving into different environments. What are the genes that underlie these traits? This is a classic question in evolutionary biology and the study of these things. We know that there are a large number of genes. This was an old review that looked at a large number of genes that are known to be involved in domestication or the movement of domestic species throughout the world and their adaptation to different environments. And we just had a recent review where we talk about some of these genes, and a lot of these genes are known. I'm just going to show you, I've been involved in the study of several of these genes. This is the gene that leads to broccoli and cauliflower in their interesting inflorescence. We know it's a mutation in a Madsbox gene transcription factor that causes loss of function of this gene in both broccoli and cauliflower that leads to that trait. Something in diversification of sexual systems. Melons are andromonaceous, and that seems to be a mutation. This is something we studied with Bendamin in INRA in France. It's a mutation in an ethylene bisynthetic gene that gives rise to the evolution of this different sexual system in melons from the wild species. An interesting case that we studied very early on, and again, this shows Nina's influence because she was the one who cloned the waxy gene in maize, which involved starch biosynthesis, and my PhD was on the waxy gene and the transposable elements in them in maize as well, and then later on in rice. And so when we started to look at rice domestication, and I was looking for a gene study to illustrate the evolutionary process, I said waxy. And the reason for waxy is that we knew from work in different labs that the stickiness of the grain of rice when you cook it is due to a mutation in the waxy gene. So different rice varieties have different levels of amylose and amylopectin. The stickiness of rice is due to the ratio of amylose and amylopectin. The greater the amylopectin in rice or branch starch, the more sticky the rice is when it's cooked. Indica rice has a high level of amylose, low level of amylopectin. It's not very sticky. Temperate japonicas have low amylose, higher amylopectin. It's stickier when you cook them. And of course, there's a class of rices called glutinous rices that have no amylose. They're all amylopectin. And this is the glutinous rice or very sticky rice. And the reason I wanted to study this is because we didn't do it, but others had figured out the mutation that makes rice sticky. And it is a mutation in the first intron donor of the spliced donor of the first intron of the waxy gene. So there's a mutation that abolishes this spliced donor site. What it does because of the abolishing of this spliced donor site is that it leads to incorrect processing of the messenger RNA of the waxy gene in individuals that have that mutation, which leads to low activity of this amylose bisynthetic protein, which leads to low amylose, which leads to sticky rice. For an evolutionary biologist, this was a great system because it allowed you to go all the way from a single mutation to a phenotype. And to try to connect all of the dots in between. So we did a large number of studies of this. I'm not going to go through all the studies, but one of the things that it allowed us to do was look at what happens when you select for a mutation that's beneficial. In this case, this is a mutation that's beneficial in a variety of cultures that prize sticky rice. So these are the Northeast Asian cultures like Japan. It turns out that if you look at variation across the genome of rice around the waxy gene where that mutation occurs, you see that there's a very small amount of variation around that region. It's about 240 kb of no variation in individuals that carry that mutation in the waxy gene. Around that, or spanning that, there's normal levels of molecular variation. And the reason you're seeing that is because of a phenomenon of a selective sweep. And the selective sweep is what happens when there is selection for a mutation that has a positive effect on the population. And the way that happens is that we know that individuals are always accumulating mutations, whether they're by SNPs or by transposable elements and so on. And so that's why individuals are different from each other. That's one of the reasons they're different from each other. But when a beneficial mutation occurs, it rises in frequency quickly in that population so that individuals that carry the mutation look very similar to each other around where that mutation occurs. And it's only broken down by recombination the further you are away from that mutation. And what that does is it leads to a region in the chromosome around a mutation that's selected for that has very low levels of nucleotide variation. And this is what's called a selective sweep. And every time you look at a genome, which is normal levels of variation, and then you see a sudden dip in variation, the size of which is dependent on how strong it's being selected or the recombination rate. Anyway, it suggests that there's something going on in that region of the genome that's under selection. And in fact, we've been using that in rice to catalog possible genomic regions that have been under selection. And so we see that, for example, in chromosome 10, there's these two regions that are about 1.5 megabases and at least one variety group that have almost no variation. So it's a large chunk of the chromosome. There's almost no variation. And one of them overlaps with a QTL, or quantitative trait locus, that's known to be involved in the domestication process. And we can use that to at least map genes, or at least localize where genes might be that are important during the domestication process. So for example, this is a region with 42 genes, about 400 kb level. You can see the region of no variation. There are 42 genes in this region involved in increasing the amount of seed that's been set, increasing yield of domestic species of rice versus wild species. This is another one that decreases the size of the panicle. And so we've been cataloging these, and we're coming up with a catalog map of selection of the rice genome. And what I'd like to end in rice is that where we are right now is we're not only taking this information about variation in the genome, we are integrating this with a knowledge of the interactions among the genes in the genome. This is the current study that we're doing, looking at the systems biology of rice. What we've done is we've taken all the genes in rice and inferred, we've done a series of time series experiments looking at drought tolerance and heat tolerance, and also taken the public database and used the model that was developed by Rich Bonneau at the NYU Center for Genomics and Systems Biology to look at the interactions of many of these transcription factors in their target genes. So this big globular map here has almost 5 million interactions that are mapping here, including 2,500 transcription factors. We're in the process of validating this. This just came out about a month ago, and we're very excited about it. You can actually zoom in and look at individual transcription factors. But what we're doing is we're trying to understand now the nature of these interactions among these genes and trying to correlate them with what we're observing in molecular diversity that we're seeing in different individuals or different populations. How is the diversity we're seeing mapping onto the networks that we're being able to infer by our experimental methods in the lab? So that's where we're going. And I'd like to end this by telling you a little bit about a project that we're doing at NYU Abu Dhabi, since we're in this region. And while rice is an important part of Saudi cuisine, date palms are even more important. Phoenix dactylifera is the major indigenous crop of the Arabian Peninsula. It's a diploid. It's about 600 megabases outcrossing. It's a perennial fruit crop, a long lifespan, a very long juvenile stage. And we have a project. There's very little known actually about date palms, about the genetics of date palms, or about their evolution. So we started a date palm genome diversity project at NYU Abu Dhabi. Our goal is to sequence 100 date palm varieties to trace the origin and evolution of these species, develop a global genetic diversity map, and try to use it to map a variety of different traits that are important in date palms. We have a project called 100 Dates, exclamation point. And our goal is to sequence 100 date varieties. We've already sequenced about 42. And we have about 10 million SNPs across the date palm genome. Our estimated sequence completion for 100 varieties is March 2014. We're only limited by the fact that we can't find varieties fast enough in the different countries in order to complete our sequencing. But what we hope to do is correlate the molecular diversity we see across the date palm genome. This is one chromosome of date palms and the diversity we observe with the large amount of diversity we see in the phenotypes of different date palm varieties in a variety of different things. And we're already beginning to glean things. For example, we've known that date palms have a very small number of disease-resistant genes compared to other species. It's at par with bananas, for example. The number of NBS, LRR disease resistance genes are lower than many other species, but the amount of variation, molecular variation in these genes are higher than the genome average because that's what we expect for disease resistance genes. We're cataloging all of these possible functional consequences of variation in the date palm genome. This is a trait that we're interested in trying to map. This is the fruit color. It turns out that there's a gradient in fruit color of date palms. Here in the Arabian Peninsula and, say, in Iran and Iraq, there's a preference or an equal preference for yellow and red dates. But if you go to North Africa, it's almost overwhelmingly yellow dates that are preferred. We think this is an easily mappable trait. So there are a large number of traits, the sugar content and so on, that we're trying to do with this project. So I gave you a very rapid and very small slice of the work that we've been doing in domesticated species. As I said previously, the reason we're interested in looking at domesticated species is we're interested in trying to understand their evolution because it might tell us something about the evolutionary process in general. But studying how these species have evolved will allow us to understand how these Neolithic farmers, these are the very earliest farmers, somehow changed the evolutionary trajectory of species to increase yield, ability to grow in agricultural environments, and in different environments from that in the wild, in a regime of changing climates. We think we can learn lessons. We don't know specifically what lessons, but clearly we can find genes that are important in many of the traits that we're interested in. And so we're hoping that this is something that comes out from this very basic scientific research problem. How do these crops evolve to something that's more practical? What are the genes underlying adaptation in plants in response to different environments? So I'd like to end by thanking my group. This is my joint group from New York and Abu Dhabi that are here. And there's only 2 people from Abu Dhabi here, Khaled Azuri and Florencia Schlam. The rest are in my New York group. Actually, Jonathan Flowers shuttles between New York and Abu Dhabi, and so with Rachel Meyer. Most of my domestication work has now moved to Abu Dhabi. These are just some of the people that have been working in various aspects of domestication. In my lab, we're domesticated species. Some of them have gone on to their faculty positions in various places. These are collaborators that have been working with me in the last few years. And of course, this work is carried out by funding from NSF, funding from NYU Abu Dhabi, and of course, I work at the Center for Genomics and Systems Biology in New York. So I'd like to thank you for your attention, and I hope you learned something from the stories I told. And I don't know if I'm supposed to take questions now, or maybe for a few questions. Thank you.
Turn it on? Yeah. Time for a few questions.
Somebody over there, at the back over there.
Okay. Can someone take a microphone back there?
I can hear you. So.
Hold on here. It's coming.
Or is the mic coming?
Thank you. Well, my question is about the selective sweeps.
Right.
So you are finding these with the SNPs,
Right.
if I understood it correctly. And I wanted to know if you're planning to detect loci in the genome that are under selective pressure in different habitats, like, for example, hotter environments, and then target these regions for, I don't know, like genetic modifications, mutations, and new lineages for certain habitats.
That's exactly the project we're doing now. That systems biology map, the interaction map, is because what we're trying to do now is we're now interested in the process not of the origin of the crop species, but how they've spread and adapted to different environments. So we're looking at drought, and we're looking at temperature, and we're probably going to look at salt stress in the future. So we're interested in how different populations are now adapted to different ecological environments. So that's exactly what we're planning to do.
Other questions? There's one over here.
Yeah, so thanks a lot for the nice talk. So these regions with the selective sweep, do you know what type of genes are found in these regions? Are they secondary metabolite, biosynthetic genes, or regulatory genes, or is it too early to say?
It's too early for many of these genes, for many of these regions. As you showed, we're able to localize the regions to about 40 to 50 genes in a region. We haven't really done any of the work to try to localize specifically where they are, what the genes are specifically. So we can't address that question right now. We're right now, frankly, just cataloging all of them. And hopefully, we'll be able to take it to looking at the single gene level and trying to find out then what are these genes? Are they transcription factors? Are they metabolism genes and so on?
Maybe a second question with the dates. So, well, we kind of did some work on dates on our own. So I'm a little bit surprised that you say there are very few kind of disease resistance genes in dates, because dates are exceptionally rich in their secondary metabolite profile. So they produce a large variety of very unusual polyphenolics, which I think would be involved in resistance.
Right. Well, maybe that's a trade-off, right? These are a special class of genes for disease resistance, the NBS LRR genes. It may be that there's been a different way by which it's doing disease resistance, especially for a perennial crop. You'd think it really would want to have a lot of disease resistance around it. It's just an observation. Maybe it's a secondary metabolism that's helping in this case. It would be an interesting hypothesis to look at.
One more question, let's move on to the next talk. One question, two microphones.
Thanks for an excellent talk. I'm actually from a medical background, and I've been very interested with regard to cancers. And plants don't seem to have cancers. Is there anything in their genetic makeup that prevents them getting cancers?
Well, no, it's not the genetic makeup, it's just the structure. The problem is that, no, it's not the problem. It's got a thick cell wall that's cellulose, and so plant cells are rigid. So you can get tumors, but you can't get cancers that spread because the plant cells are fixed because of the cell wall that's a cellulose rigid. You can't get a plant cell detach and move to another place to metastasize like you get in most cancers. You can get tumors, but they're localized, right? While cancers usually also metastasize and spread. And so it's not really the genetics, it's just the anatomy of plants in general. Cell walls are rigid, and cells are rigid and they stay in place. They don't spread, they don't move. So that's why.
Thank you, Michael. Great talk.